So the main things you want to know out of exercise 19, we'll go over in this PowerPoint. This slide is definitely important. You should know what is contained in the different fractions of blood. When you centrifuge blood down, you put the heavier elements towards the bottom and the lighter elements on top. It should make sense that red blood cells are the heaviest. They've got the hemoglobin that contains iron, which makes them very dense. So the bottom part of your formed elements are your erythrocytes. The top little buffy coat layer are the leukocytes, and above that are the platelets. So you should be able to tell me, again, formed elements being cells or parts of cells, fractions of cells, which are platelets. You should be able to tell me what is found in each of these sections. If I gave you the red cells or a different group of white cells or platelets, you should be able to tell me that's in the formed elements of the blood, and you should be able to recognize what is found in the plasma of the blood, various dissolved proteins, gases, nutrients, ions, hormones, things of that nature. So be able to tell me what is found in each fraction of blood. This is not something I would ask you in detail, but it's just to give you a feeling for where the red blood cells come from, as well as white blood cells. They all come from a hematopoietic stem cell. As you notice on the diagram on the left there, the nucleus is ejected from the red cell as it's developing. So the red cell is not technically alive. It doesn't metabolize. It's got a definite lifespan, and that's why we need red cell replacement on a constant basis. You should, again, with the leukocytes, be able to tell me what group they fall under. You should know the three granulocytes, the basophils, eosinophils, and neutrophils, and members of the agranulocyte family, the lymphocytes and monocytes. Monocytes, remember, also known as macrophages once they get into the tissue and start roaming around and consuming bacteria, dead cells, things of that nature. Again, don't confuse lymphocyte with leukocyte. Remember, leukos refers to white, and lymphocyte is a subset of your leukocytes. If it helps to remember, again, look at the right, the diagram on the right. The subsets of lymphocytes, B cells, T cells, and the NK, natural killer cells. B cells produce antibodies, and T cells communicate directly cell to cell, but if you remember, your lymphocytes create antibodies. So those are found within the fluids, the lymphs of the body. If I have you recognize leukocytes by sight, I will most likely use these photos. They are very clear and evident as what you're looking at. From left to right, you get the lymphocyte. Again, large nucleus, thin layer of cytoplasm outside of the nucleus. The basophil has got a bilobe nucleus, but you typically cannot see that for all the granules that you find within the basophil. Monocyte, slightly larger than the other four leukocytes, but the monocyte has a lobe nucleus as well, sometimes C-shaped. It just depends on what angle you see the cell at. Neutrophil has got a multi-lobe nucleus as well, anywhere between 3 and 5 nuclei. The nuclei are often described as a string of pearls. They're little globs that are attached with little bridges to each other. And the eosinophil is going to be a pink staining cell that always reminds me of somebody wearing sunglasses, because it's got this bilobe nucleus, and it is connected by a little bridge as well. So the leukocytes and erythrocytes, you should be able to tell me which one I'm talking about if I gave you the function. Obviously, the red blood cell transports oxygen, carbon dioxide to a small degree, but it's oxygen mainly that binds to the hemoglobin. Most of the carbon dioxide is actually transported, dissolved within the plasma of the cell. Neutrophils, they're phagocytic. They're also the most populous and usually the first on the scene of a localized infection. Eosinophil, these guys reduce the inflammatory response to kind of end the inflammatory response. But the big thing about eosinophils you want to know is they show up during parasitic infections. Basophils, when you look at all those granules within the basophil, they are filled with histamine and heparin, and they promote inflammation. You think when our inflammation starts to go too much and becomes uncomfortable, we take an antihistamine to reduce the amount of histamine. But histamine draws other leukocytes into the area and increases the signs of inflammation, which is redness, heat, swelling, and pain. Lymphocytes, again, the B cells are a subgroup of that, so lymphocytes produce antibodies that contribute to allergic reactions, graft rejection. They regulate the immune system as well, but antibodies come from our lymphocytes. And monocytes are a large phagocytic cell. Again, once they squeeze out of the blood between the epithelial cells and work their way into the tissues, they're known as macrophages, literally big eater. So they're eating up bacteria, dead cells, and things of that nature. And platelets are involved in clotting and release factors are involved in blood clotting as well. So with this diagram, you want to understand what's going on with the red cell. The red blood cell is what's described as a biconcave disc, where the inner part of the cell is thinner and the periphery of the cell is thick, kind of like almost like a donut, where you pinch through the donut, but you don't quite make the donut whole. You just make the center thinner. Each red blood cell is full of hemoglobin molecules, which you see on the upper right. Hemoglobin in the adult has 2 alpha and 2 beta subunits. And within each subunit, you've got the globin portion, which is what you're seeing as the gold or purple lines. Those are representing the 3-dimensional structure of the protein structure here. And the heme portion you see below, that is an organic molecule that has an iron molecule in the very center. It is this iron molecule that binds to the oxygen and carries the oxygen through the blood. So if you think each hemoglobin molecule combined 4 oxygen molecules total, 1 for each heme subgroup. And another thing to kind of be aware of is fetal hemoglobin has got different subunits and is more attractive to oxygen than adult hemoglobin. It should make sense. The baby needs to get its oxygen from mom. It's not breathing yet, so it has to have a stronger attraction towards hemoglobin than what the adult. A stronger attraction towards oxygen than what the adult hemoglobin does. So just kind of be aware of what a hematocrit is. It's a quick way of determining certain types of anemia. Basically, it's a way of determining how much red blood cells you have in your blood. It's just like the first slide we see. You take a drop of blood and you centrifuge it down. The red blood cells are packed towards one end, and you can get a rough estimate as to the percentage of red blood cells within the blood. Don't let the next few slides overwhelm you. What I want you to get out of here, the big picture type of thing, is to understand there's 2 pathways that lead to coagulation. The extrinsic pathway, which is from tissue damage, and the intrinsic pathway, which is specifically damage of blood vessels and contact with collagen. Both pathways coalesce at factor 10. And if anything, I would ask you from factor 10 down to the clot, and be able to tell me what, if I gave you the diagram below and left a few blanks out, be able to fill those in. Again, there are various terms for different structures, things like that in anatomy. So in this next slide, what you see is prothrombinase. It's going to be called prothrombin activator. But if you just remember the suffix ase is an enzyme that causes some change in whatever chemical the first part of the name is. So prothrombinase is an enzyme that activates prothrombin into thrombin. When thrombin becomes active, it turns fibrinogen, which is an inactive form of fibrin, into fibrin, and fibrin as well with the platelets forms the clot. Fibrin kind of just what it sounds like. It's fibers, and you kind of think of that as a net that catches red blood cells. So with this diagram here, again, basically the same as the one on the previous page, but again, prothrombinase is replaced by prothrombin activator. But again, it does the same thing, activates prothrombin into thrombin. Thrombin activates fibrinogen into fibrin, and then fibrin forms the clot. On the right-hand side, if you haven't remembered from the very first week, hopefully, of anatomy, when you talked about negative and positive feedback mechanisms, clotting is a positive feedback mechanism. And if you look at factor 12, it's not just one molecule of factor 12 activates one molecule of factor 11. It's, they activate several molecules of factor 11. One molecule of factor 11 will activate several molecules of factor 9, and vice versa. So during positive feedback mechanisms, it increases the change. So you get a whole lot of fibrin become active from just a few factor 12 molecules becoming active. So you want to understand blood typing and what type of blood can be donated into what type of recipient. And again, this is just a very basic understanding of blood typing. This does not even account the Rh factor. And there are other blood types out there besides A and B, and A, B, and O. Those are just the most common. So if you look at the columns, if you are a type A blood person, you have a specific antigen, type A antigen, on your red blood cells. Remember, antigens are bound to a cell. Antibodies are floating free throughout the plasma. So throughout our lives, we'll be exposed to things, and a type A person will end up making antibodies against type B blood. They're not going to make antibodies against their own antigen. They make them against foreign antigens. So likewise with the second column from the left, if you're type B, you will make antibodies against type A antigen. If you are type AB, you have both A and B antigens on the surface of your red cell, and you see both as normal, as self. So you do not make antibodies against either A or B. And if you're a type O blood person, you have no antigens on the surface of your red cell, and you make antibodies against both A and B. So they both are seen as foreign. So when you consider blood donation, you look at the antigens on the red cell of the donor, and you compare that to the antibodies that will be in the plasma of the recipient. Okay, so it should make sense that you can take O blood and put it into any blood type. All right, O blood has no antigens on the surface, so even if you put O into another O person where there's antibodies against A and B, there's no antigens on the surface of that cell to react against. So O is your universal donor. AB, if you took AB blood, and you can only put that into an AB person. If you put that into an A-type blood, you have anti-B antibodies in an A-type blood that would react against the B antigens on the surface of AB blood. If you put it into B-type person, you will have anti-A antibodies that react against the other antigens on the surface of that AB. So AB blood can only go into another AB. Likewise with A and B, you can put B-type blood into B, or you can put it into AB. You cannot put B blood into an A because the A has anti-B antibodies, and you cannot put B into an O because an O person also has anti-B antibodies. Likewise with A, you can only put A blood into an A person or into an AB person. So have a good understanding for blood typing and what type you can put into what type. So what happens if you put a type of blood into the wrong person? So at the top you have a type A blood, and you put it into another type A that has the anti-B antibodies. Well, the anti-B antibodies wants to bind to the B antigen. If you don't have the B antigen there's no binding. If you put A blood into a type B person, and that B person has anti-A antibodies, that antibody will bind to the antigen. And the fact that the antibody has two binding sites allows it to bind to two different red cells. Take that picture on the bottom right with the anti-A antibody and the A antigen on the red cells, and extrapolate that out, If you have multiple antibodies, multiple cells, you can have cross binding that just causes a great big clump that is referred to as agglutination. And if that occurs in the body, that will block capillary beds and starve downstream tissues of oxygen. We're talking about a heart attack or stroke, basically, depending on where that occurs. So what you'll see as far as testing, how you can do a quick test as a blood type, on the left there you'll see 8 droplets of blood, and each of those droplets of blood have been mixed with either an anti-A antibody serum or an anti-B antibody serum. So if you look at the top row, the anti-A has caused clumping, caused agglutination. Okay, you see those large clumps of red, that is agglutination, that is clumps of red cells. Under the anti-B, on the top column, there is no agglutination. It is a nice smooth red color, kind of like a normal blood smear that you'd look on a slide. Those cells are evenly spread out. So you can tell there's agglutination reaction against type A antibodies, so you know those cells have A antigens on the surface, but there's no agglutination with type B, so there's no A or no B type antigen on that cell. So that is a type A blood. The next one below, type B, does not react against anti-A antibodies, but it does agglutinate against anti-B antibodies. So you know those cells have the B antigen, but not the A antigen. Third one down, type AB, you have agglutination in response to anti-A and anti-B antibodies, so you know those cells have both A and B antigens on the surface of them, so that must be AB type blood. And type O blood, again, that naked red blood cell with no antigens on the surface, does not react against either anti-A or anti-B antibodies, so there is no agglutination in response to either or. And to the right is just a little diagram just reminding you what type of blood can be put into what type of recipient. So again, O is a universal donor, can be given to anybody. That's why trauma blood is usually O negative, because they don't have time to do typing and cross match, so they just give O. You can give O blood to anybody. As far as the recipients, AB is your universal recipient. AB can accept blood from anybody because, again, AB does not have antibodies floating throughout as blood against either the A or the B antigen. A you can only place back into A or AB. Likewise, B you can only place into B or AB. So that's hopefully a good idea of what to focus on. Again, focus on your blood typing and understanding that, as well as the fractions of blood and hemoglobin, and kind of understanding that, as well as your coagulation, and again, understand from factor 10 down is what I'll question you on.